So we are in the week of February 1st through the 4th, finishing up cellular level of organization this week. We'll have exam one. You can take on honor lock on your own time between now and Friday. So we probably won't finish the chapter until next class. So you might want to wait until after then to take it. So, all right. So where we left off. So we left off with vesicular transport. So we just finished up a conversation about active transport. So now we're talking about vesicles. These are just basically membranous sacs, or sacs that are made up of membrane, like the plasma membrane, and they have material in them. Okay, and so there's different kinds of transport. One form is endocytosis. And in endocytosis, it's basically information going transported in. Okay, so endo's like in. So basically, there are things on the outside of the cell. The cell pinches inward and collapses on it and brings it in, creates a vesicle. And it's active, so which means it requires energy in the form of ATP. So the contents of the cytoplasmic vesicles are going to be isolated inside the vesicle. So there are three forms of endocytosis. You have receptor-mediated endocytosis, you have pinocytosis, and phagocytosis. So in receptor-mediated endocytosis, that means there's a receptor involved. So you have some specific molecule in the extracellular fluid that just means some molecules outside the cell, right? And it's going to interact with a receptor on the membrane, and it's going to be specific for that molecule. Okay, and it's going to cause the membrane to what we call invaginate, which means basically pinch inward and fuse, and it pinches off and forms a vesicle and brings whatever was on the outside in. Then we have pinocytosis. Okay, so pino means drinking. Okay, so here you're essentially pinching in and chopping in fluid. Okay, so cell drinking. In phagocytosis, phago means eating. So here you're actually taking in matter. Okay, and this happens in certain cells like white blood cells. Why? Because they're helping you fight things in terms of immunity. All right, and so you've got the ability to take in matter that's not good for you. So related to this are Pseudopodia. These are extensions in your cytoplasm or on your cytoplasm. And they're going to surround your object.
My doctor called, but he gave me this for you.
Okay.
I don't know.
Thank you. All right. So it's going to surround your object. It's going to make the membranes fuse and form a vesicle. And the vesicle then is going to fuse with the lysosome. We learned about that last week. And they are cells that are not cells, they're organelles that destroy things, okay, and digest the contents of the vesicle, okay. All right, so just like endocytosis is bringing things into the cell, exocytosis is taking things out. All right, so it's the reverse process of endocytosis. Here you're releasing material from the cytoplasm through a vesicle. That vesicle travels from the inside of the cell to the surface of the plasma membrane, and then it fuses with it and releases the contents out. Typically, it's going to be two reasons for this. It could be it's a waste product, or it could be some secretory protein or some material that needs to be secreted to go out of the cell to some other cell. All right. So we also talk about characteristics of the membrane in terms of its potential, and it has to do with charge. So typically, the cell on the outside of the cell is going to have a relatively positive charge. Inside the cell is going to be relatively negative, okay? And that's going to be important, particularly when we get to the nervous system. And so you have a potential difference when you have difference in charge on either side of the membrane. And the transmembrane potential essentially is that difference in potential between the two sides. So in a living cell, our resting membrane potential is negative 70 millivolts, okay? So the cell, again, is negative 70 millivolts lower in charge than the outside, okay? And again, we'll talk more about that when we get to nervous system. All right. So cell membrane has these electrical chemical forces that want to move ions in and out of your cell, and your membrane kind of resists that, okay? So, and these ions that are essentially held apart by the cell, because remember there's difference in charge on either side, they'd like to connect, but they can't because of the membrane. That creates potential energy. And that cell is going to use that potential energy for different purposes. An example of that is sending information in a nerve or a neuron. Okay, so when we talk about action potentials, we'll bring this up again. All right. So another term relative to transmembrane potential is resting potential. And that just means it's the transmembrane potential in an undisturbed cell. So if the cell's at rest, it's usually somewhere between 10 millivolts and 100 millivolts. Okay, and the cell is going to use energy to keep itself in that range. Okay, so it doesn't necessarily mean much to you today, but like I said, when we get to the nervous system, it'll make more sense. All right, so we've covered the nucleus, we've covered the plasma membrane. All right, let's cover the cytoplasm. So the cytoplasm is that material between the nucleus and the cell membrane. Okay, so it's two subdivisions. We have the cytosol, which is the fluid, and then the organelles that exist inside the fluid. All right, now that fluid or cytosol has nutrients, proteins, ions, and waste products in it. The organelles all have their own functions, okay? So rather than just have all, like one organelle that does everything, you have specific organelles that have specific functions. All right. So anytime we put something together in a table, it's probably an important difference, right? It's an important thing for you to know. So differences between the cytosol that's on the inside of the cell and the extracellular fluid on the outside of the cell. So, all right. Cytosol has a high concentration of potassium ions and a low concentration of sodium ions. And the extracellular fluid is the opposite. It has a high concentration of sodium ions and a low concentration of potassium ions. Okay, and that relates back to what we learned about active transport and the sodium-potassium pump. All right, and ions being pumped against their concentration gradients. And second difference, cytosol has a high concentration of suspended proteins in it. Extracellular fluid has a low concentration of suspended proteins in it. The cytosol's got small quantities of carbohydrates and has large reserves of amino acids and lipids, okay? Whereas the extracellular fluid has small quantities of amino acids and lipids, okay? And that should make sense because the cytosol, it's where the ribosomes are. You're gonna have to make protein. You're gonna need amino acids to make protein, okay? You need lipids to make ATP, right? So your mitochondria should have those. All right. Next difference, cytosol can have inclusions and the extracellular fluid does not. Okay. All right. And we learned about those cytoplasmic inclusions on, I think, the previous page. All right. And those inclusions are masses of insoluble materials, right? So just want to remind you of that. All right. So let's go on to organelles, because they're in the cytoplasm. And they can be membranous or non-membranous. So if they're non-membranous, they don't have a membrane. And so they're directly in contact with the cytosol. So things like ribosomes, flagella, cilia, centrioles, microvilli, the cytoskeleton, they're direct contact with the cytosol. Now, the membranous organelles have a membrane that's composed of lipids, and that separates them from the cytosol. So they're going to include mitochondria, endoplasmic reticulum, Golgi apparatus, lysosomes, and peroxisomes. Okay. So these all have a membrane. So let's start with the non-membranous organelles. So the non-membranous organelles have 4 components. Got the microfilaments, the intermediate filaments, the thick filaments, and the microtubules. So just like your skeleton in your body gives you flexibility and strength, cytoskeleton gives the cell strength and flexibility. All right, so we'll start out with the smallest ones, the microfilaments. These are slender strands made of protein. Diameter is going to be less than 6 nanometers, right? Remember, nanometers 10 to the 9th meters, so pretty small. And they are made of a protein called actin. What do the microfilaments do? Well, they're going to serve as anchors for the cytoskeleton. And they anchor it to integral proteins on the cell membrane. Okay, and they give your cell additional mechanical strength. All right, so they're also going to determine how thick the cytoplasm is, how viscous it is. Okay, so whether it's more watery or whether it's thicker. Microfilaments rather, are also going to produce active movement, and they change the shape of the cell or part of the cell when they interact with myosin. Okay, and we'll see that particularly when we get into the muscular system later. All right, going a little bit larger, we get to the intermediate filaments. All right, so these are kind of in between. So these are larger than microfilaments, but smaller than thick filaments. And in diameter, 7 to 11 nanometers, so a little bit wider. Gives your cell strength and stability. Again, positions your organelles, so they're not just floating around randomly. They actually have a location based on those intermediate filaments, and stabilizes the position of your cells relative to other cells around it. Next, we have the thick filaments. All right, these are larger. They're made up of myosin. Diameter is around 15 nanometers. You only will see these in muscle cells. Okay, and because they're in muscle cells, their role is to produce powerful contractions. Okay. All right, so the microtubules then are made up of tubulin, right, which is a globular protein. They're hollow. Their diameter is even larger, 25 nanometers. Okay, and based on how the cell is remodeling itself, you may have lots of microtubules or not so many. and it's the primary component or piece of your cytoskeleton, right? It's the microtubule, the larger component, the microtubules, are your cytoskeleton's primary component. All right, so what do? The microtubules, again, give your cells strength as well as rigidity, serve as anchors for the organelles as well, just like intermediate filaments. They, when the microtubules disassemble, the cell can change its shape. And materials inside your cell can actually move along the microtubules. Okay, so they can kind of serve as bridges or roads and move materials along them. Okay. Also, they're involved during cell division. Okay, so you form your spindle apparatus, allows those chromosomes, the DNA and protein, to move to opposite ends of cells that are divided. Also, microtubules form structural components of the organelles, particularly for flagella, cilia, and centrioles. Okay, so those are going to involve microtubules. All right, so the microvilli, these are small and finger-shaped projections. Again, these have those elements in it, right? The microtubules. So, or they have cellular, they have these cytoskeletal elements in them anyway. These are projections of the cell membrane. Big thing I want you to know here is they increase surface area, okay? And that's important because cells that are involved in absorption need more surface area, because more surface area equals more absorption. So where do you find these? In your digestive tract. Okay, so your intestines, your intestinal lining is covered with microvilli. All right, moving on to the centrioles. So you typically see them in pairs near the nucleus. And they have this arrangement of 9 bundles of microtubules, and then there's 3 microtubules in the middle. So it forms kind of like a tube. Or excuse me. So it's 9 bundles, and then each bundle has 3 microtubules. Okay. So it's 27 microtubules in all. And they're involved in your formation of your mitotic spindle fibers, right? We talked about that earlier. And they actually are going to control the movement of the chromosomes when your cells divide. They also help organize your cytoskeleton. And again, we'll see how that works in cell division when we talk about mitosis. The centrosome is your cytosol, or cytoplasm rather, that surrounds your centrioles. Okay, and it's quote unquote the heart of the cytoskeleton. Okay, so the centrioles and the cytoplasm around it form what we call the centrosome. Okay, then we have the cilia. These are short, look like hairs. All right, so these have an arrangement of 9 plus 2 pairs, so not 9 plus. So all right, and typically you see them in the respiratory tracts, female genital tracts. They beat rhythmically. They move fluids or secretions across the surface of your cells. Okay. All right, the flagella. These are longer, function like whips. Also have 9 plus 2 pair arrangement. Sperm cells, the only human cells that have these, and they help move the cell through fluid, right, to get to the egg. All right. Then we have the ribosomes. All right, these are sites of protein synthesis, composed of a heavy unit, subunit, and a heavy, light ribosomal unit. So light and heavy ribosomal units. Ribosomes are composed of ribosomal RNA, ribonucleic acid, and protein. You also have, free ribosomes in the cytoplasm, right? So not three, they're freely floating, all right, as well as attached to the endoplasmic reticulum. And so we'll see later when they are attached to the endoplasmic reticulum, we call that rough endoplasmic reticulum. Okay, but ribosomes are where proteins are made. Okay, so those are the non-membranous organelles. So what I want you to do right now is to talk with your partners, or your battle buddy, and discuss 3 non-membranous organelles. All right, names and functions. All right, you can do this at home as well. All right, so now that we've covered the non-membranous organelles, let's talk about the ones that do have membranes. We'll start with the mitochondria, known as the powerhouse of the cell. So almost 100% of the production of ATP in your cells is happening at the mitochondria, okay? Has a double membrane, so 2 layers, and has cristae, which are folds in the inner membrane, okay? So when you see the mitochondria, and you see all the little folds on the inside, those are cristae. Now the contents of the fluid, we call that matrix. And the matrix has a set or its own DNA, all right? And has a separate form of RNA. So which makes it very unique to the other organelles, right? So, because most of the organelles don't have their own DNA other than the nucleus. All right. And the mitochondria can divide. They actually can divide themselves. Okay. So currently, where we think from a scientific standpoint is that we usually call like an endosymbiosis theory, where mitochondria are believed to be descendants of ancient bacteria that somehow got in a cell, and our cells essentially decided to keep them as opposed to destroy them, okay? And so there's a symbiotic relationship where both the mitochondrial descendant and the eukaryotic cell benefited. All right. So you are going to need to know a little bit about how the mitochondria produce energy. Okay, we're not going to get into every chemical reaction, but we are going to want you to understand the overview. Okay, so first part of that, we have a process called glycolysis. And anytime you see these words, what I want you to do is break them down in their components. So glyco, anybody remember what glyco means? Sugar. What does lysis mean? Breaking. So what are we doing in this process? Breaking down sugar. Very good. So we take sugar, or glucose, which is 6 carbons, break it down into 2 3-carbon molecules of pyruvic acid. All right, and the mitochondria then are going to remove a carbon dioxide molecule from each pyruvic acid. Okay, so that's what we do in glycolysis. All right, and then next phase, or next set of steps, we call that the tricarboxylic acid cycle, or TCA cycle. Sometimes you'll see Krebs cycle, KREBS. It's all the same thing. And it's a pathway which is catalyzed by enzymes. Remember those proteins that catalyze chemical reactions, and they break down organic molecules. Remember, organic means they have the carbon and hydrogen and oxygen, right? So they essentially are going to have electrons that get removed in that process, and they convert ADP to ATP. So you're making ATP, adenosine triphosphate. All right, So metabolism is just a bunch of chemical reactions, right? So if it's aerobic, it does not mean that they are doing jazzercise. It means that they are requiring oxygen. Okay, that's what aerobic means. Okay, so they're going to be breaking down these organic starting materials or substrates into carbon dioxide and water using that pyruvic acid that was made in glycolysis. Okay, but that's, if it involves oxygen, it's aerobic metabolism. All right. So that's basically what you need to know about metabolism. Okay. I mean, there's a lot more detail than that, but that's really what you need to know. All right. The other big thing there, and we'll get into this more, muscle system, is glycolysis produces a little bit of ATP, and TCA cycle produces a whole lot more. Okay, so that's why aerobic metabolism is so great. Okay. All right. Endoplasmic reticulum. So it's a bunch of membranes, and they connect to the nuclear envelope and form hollow tubes and sheets in what we call cisternae, which are round chambers. So, all right. Functions of the endoplasmic reticulum: synthesis, storage, transport, and detoxification. So for synthesis, they're making proteins, they're making lipids, making carbohydrates. For storage, they can store molecules or other materials that get absorbed from that cytosol around it, right? So then they can transport things, move materials from one place to another, and they do that through vesicles, and they can be involved in detoxification. Okay, so you can neutralize things like toxins and drugs, all right, through enzymes in the endoplasmic reticulum. Two types of endoplasmic reticulum: you have the rough ER and the smooth ER. We'll start with the smooth. Smooth ER or SER, there's no ribosomes on the surface, so it's as a smooth surface, right? And it's where lipids are synthesized, okay? And also some carbohydrates. And it's where calcium can be stored in striated muscles, okay? You need calcium so that you can have your muscle contractions, right? So that's the smooth endoplasmic reticulum. What about the rough endoplasmic reticulum? Or rough ER or RER? It has ribosomes on the surface, and that's why it's rough. And because it has ribosomes, it's where we make protein, or one of the places where we make proteins. And it can also modify those proteins through chemical reactions and then package them, okay? Then we have the Golgi apparatus. So the Golgi apparatus is going to consist of saccules, which are essentially membranous discs that are flattened. What does it do? Well, the Golgi apparatus makes and packages different secretions, things like hormones, other enzymes that get released. And that's why they're secretions. Renewing or modifying the cell membrane, and packaging special enzymes, okay, within the vesicles for use in your cytosol. All right. So what do these transfer vesicles do? They essentially move materials from one saccule to the next saccule, okay? So on one organelle, from one organelle to another organelle. So there's actually 3 different kinds of vesicles. All right, we got vesicles that are involved with secretions, and they eject those secretions from the cell using exocytosis. Remember, exo means out. We talked about that already. New membrane components. You're going to have the Golgi apparatus changing the properties of the cell over time. Okay. So there's adding new things to the membrane, basically, is what that's saying. All right. And then packaging enzymes inside the cell. So example of that are lysosomes, which are essentially vesicles, okay, that have digestive enzymes in them. They break stuff down, right? Destroy things. So speaking of lysosomes, let's talk about those. So they're vesicles, again, they have digestive enzymes. They're made by the Golgi apparatus. If they have inactive enzymes, they're primary lysosomes. If their enzymes have been activated, then they're considered secondary lysosomes. So what do? Well, the lysosomes are going to fuse with other vesicles. And then those vesicles are going to get released onto whatever was in that vesicle and digest them. Can also fuse with organelles that have been worn out and digest those organelles. Call that autophagy, because since it's kind of your self-eating, all right? But that's a way to recycle parts that aren't working anymore. All right. Lysosomes can also help you fight disease, can break down bacteria that your cells have ingested through endocytosis. And then they brought them into the cell. They're generally involved in cleanup and recycling. Basically, they also have a function called autolysis, where it's called programmed cell death, where cells that are basically in bad shape, there's signals that are released that your lysosomes will just basically destroy them. Okay, and this can happen like with cancer and other types of situations. All right, and also just cells have a certain number of divisions. So after a certain number of times they divided, they tend not to work optimally anymore, and they can go through this process. All right, so the peroxisomes, they're going to carry their enzymes. They're absorbing and neutralizing toxins. Basically means they bring them in and they make them not dangerous, all right? Like alcohol and hydrogen peroxide, okay? And they protect your body from chemicals called free radicals that cause chains of chemical reactions. And so that hydrogen peroxide that it's absorbing has to do with why it's called a peroxisome. All right. And these membranous organelles are either interconnected or they can communicate with each other via vesicles. Okay. All right. And this movement and exchange of vesicles is called membrane flow. All right. So those are the membranous organelles. So what I want you to do is talk with your battle buddy about each of you should discuss the names and functions of 3 different membranous organelles. You can do this at home as well. So now that we talked about the cytoplasm, let's talk about in the organelles, let's talk about the nucleus. So that's your control center. All your cellular operations are controlled from there. All right. So basically, all your structural and functional characteristics are going to be determined there by which proteins end up getting synthesized and then how much of them get synthesized. So your nucleus has an envelope that surrounds it. It's a double membrane, 2 layers, and it has a perinuclear space. All right, it has a little nucleus, little space in between the membranes. All right. So you also have pores in the membrane. These are holes or nuclear envelope. And that allows for communication between the nucleus and the cytosol through those openings. Okay, so chemicals can get through. So just like the cytoplasm was where the fluid inside the cell was, the nucleoplasm is where the fluid inside the nucleus is. All right, and then you have nuclear matrix, and it's a network of fine filaments that are inside your nucleus. Okay, it's going to provide structural support. So your chromosomes, this is where you take your DNA and you wrap it around these histone proteins. Okay, that's what chromosomes are. Okay, and that's good because when your cells get ready to divide, it's easier to grab the DNA if they're, clumped up together, as opposed to just floating everywhere. All right, the nucleosome is essentially a complex of DNA strands that are wound around histone proteins. Okay, now your chromatin is basically what happens in non-dividing cells. So your chromosomes are tangled up in these filaments. So that's what you have when your cell is not dividing. When your cell is dividing, it forms the chromosomes. Okay, so chromatin is when your cell, the DNA when your cells are not dividing. Chromosomes are when they are dividing. All right, so the nucleoli, it's plural. Nucleolus is singular. Okay, this is space inside your nucleus where our ribosomal RNA is made. Okay, so this is where essentially your ribosomal subunits are made, or at least the RNA part. Okay, genetic code. No, that's on your review sheet. So your genetic code is basically the method how information gets stored in your strands of your DNA inside your nucleus. Okay, and it's a triplet code. So you want to know that term. It's a triplet code. And it's highlighted or it's underlined, so you probably want to know it. Because each amino acid is identified by a sequence of 3 nitrogenous bases in your DNA. Okay, so every 3 nitrogenous bases signals for a specific amino acid. And each gene has all the triplets it needs to form a specific protein or polypeptide chain. All right, and every gene has segments that are going to regulate the activity of that same gene in the protein that it makes. So just like your fingerprints kind of are markers that tell you, identify people, you have DNA fingerprints, and these are essentially based on how many repeats of certain sections of your DNA you have. And that can be a marker for different genes, different people, things like that. All right, this is real critical right here. I want to make sure y'all understand this. So before genes get activated to begin protein synthesis, RNA polymerase has to bind to that gene on the DNA, right? So an ACE, anytime you see ASE, it's an enzyme. So RNA polymerase is an enzyme that makes RNA polymers, okay? So protein synthesis occurs through 2 main steps, transcription and translation. So transcription is how you're transcribing DNA or decoding it, and you're using that to make RNA. Translation is how you make protein. Okay, I want to be clear. DNA never becomes RNA. All right, everybody say it with me. DNA never becomes RNA. RNA never becomes protein. RNA never becomes protein. Okay, DNA is used to make RNA. DNA is used to make RNA. RNA is used to make protein. RNA is used to make protein. Okay, so These are processes that you use to make things. And it's DNA to RNA, or DNA using DNA to make RNA is transcription, because you're essentially using the same language. You're just creating a transcript of that, okay? Translation means you're going to a different language, right? So, because DNA and RNA are nucleic acids, it's the same language. Protein's a different language from nucleic acids, okay? So that's translation. So let's talk a little more about transcription. So in order to do that, we need to understand the RNA. And there's 3 different types. So you have messenger RNA, transfer RNA, and ribosomal RNA. So in messenger RNA, so this is your linear sequence of RNA that is essentially decoded from the DNA. Okay, and that's what goes to attach to a ribosome in the cytoplasm. Your transfer RNA looks kind of like a clover, okay? And it actually picks up amino acids in your cytoplasm, okay? And your ribosomal RNA makes up your ribosomes. Okay, it's got that heavy subunit and the light subunit. You need all 3 to make your proteins, okay? So essentially you need a site, which is ribosomal RNA. You need a sequence, which is messenger RNA, and you need something that brings the components, which is the transfer RNA. All right, so 2 strands that you need to make sure you understand what they are and how they're different. Your coding strands, so your DNA, if you remember, it's double-stranded, right? DNA has 2 strands. So one of those strands is a coding strand, all right? And it means it has the triplets that are specifying your sequence of amino acids, okay? Now you also have a template strand, which is one of your strands of DNA. It's containing the complementary triplets that will be used as a template to make your mRNA. All right, so your mRNA, it's going to have a nucleotide sequence identical to that of your coding strand. The exception is that RNA has uracil, DNA has thymine. So all right, this is a template. All right, man. So complementary base pairing rules, A binds with T, C binds with G, T binds with A, G binds with C, C binds with G, A binds with T, or base pairs, A base pairs with T, G base pairs with C, C base pairs with G, okay? All right. Now, the mRNA strand you're gonna make in transcription, okay, this is your template strand, so you're using the template to make your mRNA. And it also follows the base pairing rules. So T is T base pairs with A, G base pairs with C, A base pairs with not T, but U, right? RNA, no thymine. G base pairs with C. Well, C's base pairing, or G's base pairing with C, T base pairs with A, T base pairs with A, C base pairs with G, C. All right. What do you notice about the mRNA strand and the coding strand? That's the only difference, was the uracil, right? That's what I was trying to say. I felt like I could show it to you better than I could tell you, right? So that's what that means. All right. So codons, yeah, we'll go here. So it's a 3 nitrogenous base sequence in your mRNA. And it's going to match up with an anticodon in your tRNA. All right, so these are all codons. Okay, 3 nitrogenous base sequence. So these are codons in the mRNA. In the DNA, or the DNA and the RNA, we call these triplets. All right. But your tRNA essentially is going to look like I said, looks like a clover. It's going to bring over, and it's going to have its own matching sequence. All right, this is your anticodon for the tRNA. Okay. All right, so transcription. Make RNA from a DNA template. So after mRNA transcription, your mRNA strand is going to carry its instructions from the nucleus to the cytoplasm. It's going to go through a nuclear pore. So what are the steps? And we'll finish with this. First step, you're going to unwind your DNA apart so you can make a transcript. RNA polymerase, that enzyme, is going to attach to your template strand. So you're going to separate these two. The RNA polymerase attaches to the template. The nucleotides are going to start attaching to your template strand according to these bonding rules, right? Then you get a stop signal, which means no more transcription. Then you start processing your RNA, okay? Because actually some of the RNA that's made, you don't end up really needing, okay? So you cut out the pieces you don't need. All right, so the introns are taken out, and then the exons are spliced or are basically stitched together, all right, or glued together. Then the mRNA is ready to leave the nucleus through a nuclear pore. Okay, so let's go ahead on your exit tickets. Because this is critical, and this is actually one of those potential essay questions, for your exit ticket, I want you to explain steps of transcription, okay? All right, so that'll be your exit ticket, and then we'll pick up with translation on Thursday. That way we'll definitely finish up so you'll have everything you need for the test.